Okay, perfect. So good morning, good afternoon, good evening, everyone that's taking part in our workshop in tandem with Agrisera. I'm John. I'm here with my colleague Will, who's head of the supply team. I'm the product manager, and we work with Agrisera to distribute their products within the UK, including their Western blotting reagents. So that includes the things you'll be hearing about today in the workshop. At 2B Scientific, we also have our 2B Found system, where if you come to us requesting an Agrisera product and something either upstream or downstream within that workflow, then we can go either within our field of supplies or even beyond that to be able to get those products for you. If there are any questions throughout, please just drop them in the chat. I think, depending on Joanna's preference, you can also just ask between slides. But if it's for a distributor, it might take us time, us as the distributor, to get you some answers, depending on whether it's for the sales team, supply team, and so on. There'll be a quiz at the end of the presentation, so take part in that if you so wish. And from this point, I'll leave you in the hands of Joanna, who is the technical support manager at Agrisera. I hope you enjoy the workshop.
All right, welcome everyone. And yes, you can, I think that it will be easier if you have any questions that you type them in a chat box, and I will address them in the end of my presentation. I will focus today on a topic of interpretation of Western blot results. And I will cover some aspects of this exciting technique of Western blotting. The technique is quite complex. I'm focusing on the outcome today, but there will be a possibility to join the webinars in the future if you want to have a little bit more tips about troubleshooting the whole procedure. So let's start with a little bit of information about where we are. So it is Scandinavia, Sweden, and we have been established 40 years ago. So we have quite many years of experience in antibody production. You see here, we have two facilities, what you see on these pictures. One is taken from above our antibody production farm. And then we have another facility in the city of Umeå, from where we are doing purifications, quality control, labeling, antibody labeling, and we are shipping them all around the world. Agrisera has grown since being a small family company. We have been acquired by Olink Proteomics, and we have become a part of Thermo Fisher family last year. We're also ISO certified. And over the years, plant antibody collection has been around for 25 years. And this year we are celebrating the award which we have received from a UK-based company called Site AB, as the most publications in plant science are using Agrisera antibodies. And it's around 800 to 1,000 papers per year with our products. So we're doing antibodies, producing antibodies to plant proteins, but not only that, we also do antibodies for hormones. We have quite a vast collection of epitope tag antibodies, including Turbo ID, antibodies to GFP, HIS, these most common ones, but also antibodies to very unusual epitope tags, high titer secondaries, and we are cited in over 10,000 publications. We are very proud of it. But now we are jumping into the topic, of course, who is doing the job in Western blot and visualizing a target protein in a complex protein mix is an antibody. And on this slide, just to refresh your memory, it is a Y-shaped molecule, and it has a so-called FC region, is that one, the handle. It's called like that because it's fragment that crystallized. During research on the beginnings, that part crystallized in the fridge, and therefore the name. What is the most important and interesting for us is the antigen binding domain here. This is a variable region. This is where the antibody is binding to desired target protein. And this is how the antibodies in mammals are looking. Most of them anyhow, not in llama. In llama they are shorter, and which you're quite probably using in your research. You will receive this presentation afterwards if you would like to, as PDF, and we will also share the link to the recording. So therefore, the slides contain a lot of links as well. And we need to refresh our memory on the term epitope, because this is where the antibody is binding. And this is that part here. An epitope can be, as you see here, a subsequent sequence of amino acids in linear structure of the protein, but this epitope is called linear epitope, and it can have 2 to 15 amino acids. And this is crucial to remember because for understanding and for interpretation of the results, the antibodies are binding to these two types of the epitopes. And this type of epitope antibodies are binding to in techniques where protein is denatured, unfolded, which are Western blot in the denatured conditions, immunohistochemistry. But antibodies can also bind to conformational epitopes. As you see here, it is distant amino acids can be brought together by protein fold, and this will also create an epitope. And such epitopes are recognized by antibodies in techniques like Western blot in native conditions, immunoprecipitation, and also CHEAP, of course. And please keep it in mind that if the antibody is monoclonal, single, it means it will recognize only one such epitope, 2 to 15 amino acids long. If antibody is polyclonal, poly means many, it will recognize a pool of epitopes. I don't want to comment here which type of the antibody is better. Each of them has advantages and disadvantages of the use. And before you do any Western blot experiments, you need to make sure that you have the right antibody. What do I mean with that? Depending which species you are working with, it can be also bacterial cells, it can be human cells, it does not matter. But you need to first check that the antibody is made to a sequence which is actually existing in a protein from a species from which you are extracting it. And also, instead of doing experiments, we can also predict if a given antibody can work in a given application. Because if it is made to a protein fragment, peptide, which is buried in a protein fold, such antibody is not going to work in native techniques. So instead of doing experiments, you should first secure that you have right antibody for your application. This saves a lot of time and secures that you're using proper reagents. Each year, I'm finding few publications which are using our antibodies on species where the sequence in a protein in that species, the sequence is not even existing, but the band is published. So this is very easy to obtain false positive results with antibodies. Therefore, before you start using any antibody, you should check if it is a proper antibody just for your species and application. Therefore, Agrisera product info sheets are listing species with confirmed reactivity of a given antibody, which means it's proven experimentally, and also predicted, which is based on a sequence analysis. If you cannot find such information, you should contact supplier and make sure that you're using a correct reagent. And how do I choose the right antibody? This is what I have been talking about. If the antibody is made, we cannot assume this is made to a whole protein. It can be made to a short peptide, as you see here. A signal peptide is cleaved off, that's the bold part, and then the peptide the antibody is made to can be derived from N-terminal, from the inside of a protein, buried inside of a protein, or from the very C-terminus. It depends upon the protein. And that sequence, marked here in green, needs to be found in the protein you are aiming to detect. Of course, this is just an example for a PSBA protein, but I'm talking here in general that if a sequence antibody is made to is not shown on the product info sheet, you need to contact the supplier to make sure that you're using correct antibody. 70% of this sequence of subsequent amino acids needs to be found also in the protein you are aiming to visualize to give an antibody possibility to work. Of course, here we can only consider linear epitopes. There is no way at the moment to predict those conformational epitopes. For those of you who would like to know more, there are more links in the presentation which you can check later. You need to ask yourself, you chosen the right antibody, and now you conducted a series of experiments, but before you do it, you need to ask yourself, how much do you know about the protein you're aiming to visualize, to detect? To facilitate this binding, we have our target protein on a membrane in the Western blot in a complex mix, and we want the antibody to recognize and to bind. And ask yourself, this is your protein checklist. You need to know molecular weight of the protein, which is calculated, can be different actually. Calculated and apparent can differ. I will show you how. You need to also check isoelectric point, protein solubility, expression level, and protein abundance you can check in this wonderful database called Protein Abundance Database. Because there, for many species, there is data where you can see is your target protein you are aiming to visualize, what is its part per million level in a given tissue. For proteins of very low abundance, below 10 parts per million, it is a sign that it will be more difficult to visualize them. And there are certain precautions then have to be taken. Then also, you need to check if you are using a proper organ, tissue, and also for proteins of low abundance, a cellular localization is important because it will give you a clue if you should do fractionation. This will be very helpful when working with proteins of low expression. And now we went through all this. We choose the correct antibody. We have a proper cell extract. We know abundance level of our protein, and now comes the truth, the Western blot. And here you can see results from 4 different laboratories. The material was the same, Arabidopsis thaliana. Each lab received the same primary antibody, but as you see here, the outcome was very different. From no signal to a lot of background to one lab which managed to visualize the target protein. So this is they use different protocols. Western blot is a technique with many variables, and therefore the outcome can vary depending upon the lab. I mentioned that first on your checklist needs to be information about calculated and apparent molecular weight. And then here I will show you an example of photosystem 1 protein PSA-A. And in this case, length is 751 amino acids, calculated molecular weight was 83, but apparent molecular weight is around 55 kilodaltons. And this is very important information in your interpretation of Western blot results, because you may be confused that suddenly the protein doesn't appear at the correct molecular weight. And there are different reasons why this can happen. So what is the reason? Processing, of course, signal peptide is cleaved off, and therefore the protein migrates a different molecular weight. post-translational modifications may also influence migration of your target protein. And then down the list, we have also amino acid composition. In this case, protein is quite hydrophobic. This can also change mobility. Of course, aggregation or proteolytic cleavage, larger epitope tags, which also will influence mobility, and SDS page conditions. So these factors can influence that the molecular weight recorded on a blot will not be the same as calculated from the sequence. Here, I would like to provide you just with a checklist how post-translational modifications can influence the mobility of the protein, because depending if it is phosphorylation, glucosylation, ubiquitination, the protein may migrate faster or slower on a gel. So this is a slide for your future reference. I would like to also stress here that you need to use really good molecular weight markers. My favorite are markers with IgG binding site. And here I would like to show you an example of magic marks, which we are mixing with these colored markers to track gel progress of gel electrophoresis and see the migration of the proteins in a gel. The most common mistake which is happening is that for low molecular weight proteins, electrophoresis is conducted too long, and then the target protein is simply leaving the gel, and therefore lack of a signal later in Western blot. But here we are focusing on using proper markers, and these markers with IgG binding site will allow visualization of molecular weight directly during development of your blot. This is a very good, very neat procedure. I've been troubleshooting results where markers were assigned wrongly, the membrane was put the other way, and so on. You can avoid these kind of situations when you use these kind of markers. So markers with IgG binding site. And these markers are compatible with different detection systems, and they are making it very easy to show a proper molecular weight on your gel and help you with the analysis. Of course, in Western blot, we want to use some kind of loading and normalization controls. And what I see when I troubleshoot Western blots is that many labs are still loading on volume. And this is something which you should avoid, because if you're extracting multiple samples, then it will be a variation, a factor of your hand being tired if you're doing it manually, and the comparisons are going not to be reliable. So first of all, load on volume. And second parameter is that the signal which you're recording, depending upon your detection method, needs to be in linear range of detection. And here an example of titration. We have two blots. Upper blot has load protein per well is 4.5 microgram, and at the bottom we have 10 microgram per well. And then we are titrating the primary antibody dilution, which is used in different primary antibody used in different dilutions, and that way you can titrate where you will have the most optimal and linear detection range. Of course, it will also depend what detection reagent you are using, but please measure protein concentration in your samples and load based on the protein, not on the volume. And from the normalization controls, I would like to show you an example here. Then we have a target protein detected, high molecular weight protein, so it's quite challenging for detection because the transfer can be in a need of optimization. Larger proteins are migrating from the gel a little bit slower. They require membrane with a bigger pore size. So here we have a target, then we have a mutant, no target detected, and we have a normalization, we have a control, MPK6, but as you see here, exposure time is the same, but each of the antibodies is used in a different dilution. So this has to be also optimized to be able to be in a proper linear detection range for both targets. Crucial points here that we need to have an equal protein load per well. The bands cannot be distorted. We need to have linear detection range, and housekeeping proteins cannot be arbitrarily chosen for your experiment. It has to be experimentally confirmed that a given housekeeping protein is suitable as a loading control. We have linear detection range combined with equal protein load per well, and here I want to show you the example where two sensitive detection reagent was used for the photosynthetic protein of high abundance, PSBA, and if a reagent with extreme low femtogram detection range was applied, of course, this is not a linear detection, we get a lot of background, therefore you need to fit the proper detection reagent depending upon how much of your target protein is on the membrane for visualization. And from loading controls, you will probably recognize those names because these proteins are most commonly used as loading controls. Although, please keep in mind that they are not always suitable for your experimental setup. And I will show such an example shortly. Of course, the purpose is assessment of the equal loading per well, and depending upon the experimental conditions, this can actually be a situation when actin was not suitable as a loading control. So as you see here, in a metal stress, actin levels were actually not the same depending upon the sample. So please remember, loading controls cannot be chosen arbitrarily. You need to have a confirmation experimentally that they are suitable for your setup. Here is a list for your future reference of other possible internal controls for your experiments. Of course, we are after the ideal Western blot results when band of correct molecular weight is gone in the deletion mutant and is present in the overexpressor, is enhanced in the overexpressor. And here, this is such a ideal result, which I'm troubleshooting very many results obtained with Agrisera antibodies, which are under validation process, because we're doing it in collaboration with other laboratories. And of course, this is the most desired outcome. Unfortunately, it is not always happening. And sometimes Western blot results can be misleading. And I would like to show you this example where we have a beautiful band which is having quite nice intensity. The protein is photosynthetic TOC34, and somehow the lab missed that it migrates at improper molecular weight. So what is detected here is probably Rubisco, not TOC34. So you need to be very careful when you're analyzing your results to be sure that the band which is detected is of proper molecular weight, and you need to include all the details regarding potential protein modification, which can change mobility of your target protein. And why antibody detecting a recombinant protein target is not, why the detection of recombinant protein cannot be a confirmation that the antibody is going to recognize endogenous protein. This is actually a very common situation, and some vendors are using validation using recombinant proteins as on their product info sheets. And this is misleading because an antibody may actually recognize recombinant protein, but not a protein, an endogenous protein. There is much more of recombinant protein often present in a sample, as you will see in this example. So we have endogenous sample here, and then we have the same target protein present in the transgenic material. So as you see, intensity is not the same. And one should not even compare it, because the level of that protein in the endogenous tissue is incomparable with what you will achieve in your transgenic material. So please remember, when you're purchasing antibodies, be very careful and check if the vendor is only showing the data of detection of recombinant target. This is not a proof that the antibody is going to work on your endogenous protein. And this is very misleading. And in some cases, it can be opposite way around, that the antibody may recognize endogenous protein, but not recombinant. This has something to do with exposure of the epitope exposure, which may be different in recombinant protein, and therefore the antibody, because of a partial fold, which is still possible even in Western blot in denatured conditions, and this partial fold of recombinant protein may make it impossible for the antibody to bind. So please keep in mind that the data of validation of antibodies which you are purchasing need to be performed on endogenous samples. And if you are purchasing the antibody which is validated only on recombinant protein, it may not work on endogenous extract. This is a very, very important point for you for the future. Sometimes antibodies are, as I mentioned, not recognizing the proteins. Here we have the wild type. This is a PEP protein, so plasma membrane protein in maize. And as you see, there is a very nice band of target in the wild type. However, here we have a transgenic material, and that antibody should recognize a band around 54 kilodaltons, but this pattern is rather showing that the antibody is not recognizing recombinant target. So such situation is possible. Maybe addition of denaturant like urea could help to make it more accessible for the antibody to bind. But in the setup which was conducted in this experiment, it was not possible. So here the antibody detects only endogenous protein, not a recombinant one, which should be up here. Target protein band is not gone in knockout mutant. This is a nightmare, and that actually happens, unfortunately, in quite many of the situations, and there are several reasons for this. So in this blot, which I will show you here, we will have a wild type, and we have a wild type here, and then we have a mutant. And the band of PIF7 is unfortunately present in the mutants. Well, this is a situation where I'm not discarding such an antibody directly, because there can be different reasons for that. There are limitations in using mutants because there can be off-target binding. You need to keep in mind that this band contains not only your target protein, but hundreds of other proteins, and there can be some cross-reactivity. Of course, there can be an incomplete mutant. It has happened that sometimes mutants were wrong, and therefore the antibody was recognizing a band in a mutant. We can have some gene compensation, technical artifacts, presence of isoforms. So before I discard such an antibody, I will double check with another laboratory having a mutant, also with a protocol of adsorption, trying to see if there is any possibility to solve this issue. Another protein present in the band may actually share the epitopes with your target, and therefore the binding may occur. So this is a reason for the detection of the target protein band in a mutant. However, we don't know. In a mutant, the band should be gone, and sometimes it is not. What we can do, as I said, we can run an adsorption protocol. So we can apply an antibody on the blot where there is, which is depleted of the of our target protein, and then use this antibody solution again on your on our blot with the samples which we want to analyze. It is described in more details on our website. When we don't have mutants, what can we do? How can we analyze if the antibody is binding to the correct target protein? There are different other approaches. For example, this one, which is fractionation. Some mutants are lethal. Some transformations are not possible to be done, and therefore we can use this approach instead. So here target protein is of 37 kilodaltons, and the first sample we are trying to detect, visualize an enzyme, which comes from PTOX. It comes from thylakoid membrane. It's a thylakoid membrane bound enzyme. So sample 1 is a chloroplast. Then we have sample 2, which is thylakoid membrane. That pattern is correct. And then we take a negative sample. This is a stroma sample, and here the band is gone. So when we don't have access to mutants, we can use all different combinations of controls to confirm that the antibody is binding to a correct target protein. So we can, for example, take conditions which are down-regulating or up-regulating our target protein and check if the pattern of detection follows published data. We can also use IP combined with mass spec. Some band may be also, same band may be also recognized with 2 independent antibodies. However, we not always have a luxury of having 2 independent antibodies for the same target. And also another possibility is a multi-species detection. We are working with protein to plant targets. So here I would like to show you a so-called garden blot, which is a collection of samples from 11 different organisms, photosynthetic organisms. There are higher plants here, cyanobacteria and diatoms, and the same target protein is recognized in all of them, but as you see here with different intensity, even though a short synthetic peptide, which was used to develop this antibody, is perfectly conserved in all the species. But this type of detection can be also an indirect method of confirmation that antibody is binding to the correct target protein. How to be sure that an antibody is binding to the correct target protein? Here is the checklist for you. So first of all, we need to be sure that we are detecting correct molecular weight, and that molecular weight, calculated and apparent molecular weight, can be different between depending upon the protein and depending upon its modifications. Then we need to ask ourselves if our Western blot protocol was optimized. Do we have negative and positive controls which are behaving as they should? Do we have a linear detection range? And we can use also other approaches like IPMS and functional assays. Can we include them? And very important questions, if obtained results are reproducible. And this is our checklist for securing that the antibody is binding to the correct target protein. And as you know, in some cases, we may have such outcome. There is no band whatsoever, and there can be different reasons for it. For example, extraction method. So as you see in this example, which was sent from one of our test labs, depending upon which extraction buffer was applied, target protein band was detected. So you should never discard an antibody directly if it doesn't give you desired outcome. Another example here, we have 3 species, barley, Arabidopsis, and rice, and target protein was detected only in Arabidopsis. The sequence is conserved in all 3 species. And what happened in this experiment is that 6 molar urea was included to extraction buffer, which allowed to unfold the target protein and allow an antibody to bind. So, in case there is no band, please don't panic. Analyze your buffer composition, ask and check if you have used proper organ, if you included protease inhibitors. For some proteins, even proteasome inhibitors are important because some proteins can be also targets of proteasomal degradation. There was a lab was trying to detect a protein for over 1 year. It was a very important project, and there was no band whatsoever until a proteasome inhibitor was included. You need to optimize your extraction method for a very good yield, especially if you work with proteins of low expression. And sometimes you need to use specific conditions when you are extracting proteins. For example, do it almost in the darkness or process your extraction in a very, very prompt manner, because some proteins with lengthy extractions, even in presence of the inhibitors, then may be subjected to degradation. So protein extraction is one of the key to your success, to have bands for on your desired bands on your Western blot. So antibody is not always to blame, as you could see in this example. And if we have a band of incorrect molecular weight, there can be also different reasons for that. Let me go back to that slide. I was too quick. It can also be that there was too little of your target protein, and therefore antibody bound to something else. So I would not discard such an antibody too quickly. I would go back to the beginning and analyze beginning of the Western blot protocol, which is protein extraction, but also gel electrophoresis. Especially for proteins of low abundance, it can be quite challenging. So you need to make sure that proper protocol was applied, and there is no one protocol fits them all when working with antibodies. And we have also another situation that we may have too many bands. In this case, target protein is detected, but as you see here, it's quite a lot of other things. And this result can be still optimized because for some protein antibody pairs, incubation of a primary antibody overnight may lead to increased background signal. So you need to plan your experiments and incubate only 1 hour room temperature, your primary antibody. Blocking can be done overnight if you need that time and complete your work that day. But please remember that extended incubations of primary antibodies may increase your background signal. I also received this question: when we have a lot of cross-reacting bands, will knowing the antigen sequence will it help to identify cross-reacting bands? Now, one would think so, but actually the peptide sequence which is chosen to produce given antibody is absolutely unique, and it should not be found in other proteins. And as you know, there's hundreds of proteins in a given band. So knowing the sequence will actually not help you. It is only mass spec combined with sequencing which can tell you the identity of the cross-reacting band. So please don't go that way, because sequence analysis will not be helpful in that case. So what to do next? If something does not work, you need to go back to the checklist. Am I using correct sample? Maybe if protein is of low expression, fractionation is necessary to perform. We need to sometimes increase protein load per well. So sometimes it can be up to 50, 100 microgram and more for proteins of low abundance. Transfer conditions, sometimes with low molecular weight proteins, they may be blasted through the membrane, while for high molecular weight proteins, the transfer time may be required to be a bit longer. So you need to consider these parameters. And also using, in general, primary antibodies which are purified is of benefit. And also, of course, a sensitivity of your detection reagent. So analyze applied protocol. The most common mistake is generation of a lot of changes without the theoretical analysis. So sometimes the labs are sending results to me after 10, 15 blots, while the analysis could be done after first blot, when it was no correct band detected. So before adjusting the conditions, please do the theoretical analysis first. And I hope this presentation will help you to do it in the future. So have you experienced poor results? Wrong molecular weight of your target protein? Maybe you're struggling with lack of reproducibility, and you maybe have worse result with each blot. This is what I have seen. And what we are doing in our lab is consistent reporting of results, and we are doing it using this template where we will compare these parameters between each blots to find out what can affect the outcome. This template is always sent to our test labs, and we want to provide very many details of the protocol to help laboratories using our antibodies which are approved, which are validated, to know which conditions are appropriate. Because there is no one Western blot protocol which will fit all antibodies. This, as you see marked in yellow, are those most important parameters, and they depend actually upon protein antibody pair. They can be very specific, and therefore you will find them on our product information sheets. So when we are validating antibodies, we can have these direct approaches, reliable knockout or knockdown mutants. If you are ordering custom antibody production, an ELISA is not a method of proper validation of the antibody. We need to have some better proofs if antibody is recognizing correct target. So immunoprecipitation combined with MS or MS analysis of visualized band. And then we have, in cases when we cannot produce the mutants, we have a whole list of indirect approaches, which can be combined together. All in all, antibody validation, and as you know, Western blot can be quite challenging. And therefore, we created those educational resources. These are educational posters. One is focused on protein extraction. The other one is focused on Western blot technique. And in that poster, I try to summarize quite a lot of topics which are concerning Western blot and succeeding with it. And if you would like to have these posters, you can download them from our website or request hard copies. They are very easy to transport, and we are offering them to everybody who is interested. There are also educational posters on other topics like photosynthesis and epigenetics, and you're welcome to take a look if you are interested. Also, images from our posters are available for free download on our website. And we are also offering you 150 free antibodies in return for results. In case they are fitting your experimental questions, you are welcome to request them. As you see here, our antibodies are lyophilized and they are shipped in ambient temperature, which is more environment friendly and also good for antibody stability. If you want to check the list of these antibodies, you can find it following the link. And for all participants of this webinar, we offer 10% discount on your next order, which is to be placed with To Be Scientific, which is Agrisera exclusive distributor in UK. And with this picture, without the snow, we still have beautiful autumn here, although without the leaves. I would like to thank you for your participation. And if you have any questions, now it's time to ask them. I also would like to mention that if you would like to receive this material, so link to the recording and the presentation in PDF, please contact us. I'm not certain that we will have all your email addresses, and therefore I am providing you with my email address through the chat. And I will also send you a link to the quiz, which you can calmly do later until tomorrow. And then the winner of the quiz, who answered most questions or all correctly, can choose a free antibody from our catalog. So thank you very much for today. And now it's the time to ask questions.
If there's no questions at the moment, I'll just chime in to say, oh, apologies, we do have a question. I'll just quickly say as well, if there's any questions regarding distribution or anything regarding getting a hold of Agrisera products, especially within the United Kingdom, please feel free to reach out to Be Scientific with the email that was aforementioned on the slide prior to this one. We're welcome to answer any questions at all and help you with whatever application you might have.
Okay, I see a question. Am I correct that you said that blotting time and electrophoresis time could increase background noise? In a way, yes, because if you don't, the background noise will come also if you have too little target on your membrane for the antibody to bind. So if your blotting time is, for example, too short, you will not have enough target on the membrane, because Western blot is about signal to noise ratio. So you have to have enough target, so it will come within maybe 30 seconds, 1 minute, and then signal to noise ratio will allow prompt visualization of your target protein. So therefore, I was trying to show you that you need to also look at the conditions of your procedure. So if you have proteins of low molecular weight, you need to take care of blotting not being too long. If you have proteins of high molecular weight, the blotting should be longer. And also incubation prolonged incubation with primary antibody can contribute to increase background. I hope that I answered your question. If there are no more questions, remember you can always send them to me later by email. My email address is here in the chat. And also I see that John typed the form for the quiz. So you're welcome to take part and check your knowledge. And in case of any future questions about this technique, I'm always keen to help. And in some cases, I troubleshoot antibodies which are not produced by Agrisera or which are custom made. So you're always welcome to come to Agrisera for support. And thank you very much that you wanted to join today. And I hope that material has been useful for you.
Thank you very much, Joanna, and thank you everybody for attending as well.
Sorry, just to interrupt, John, I think there might be a couple more questions pending. I can see a couple of people are typing.
Oh, I do apologise.
I see that too.
All good.
Yeah, you're very welcome. If you have no more questions, we can close down the session and you will receive the link to the recording and the slides. So thank you very much and good luck with your Western blots.